get more questions so that I can answer to all the questions. Thank you once again opportunity to given to me in the ISPPT committee and Dr. Sriram. So let me start the presentation. So this really talk about the biosimilars. So I will I know this is topic and I don't want to give all the details. I go into the same way. So then what are the biosimilars and can you go to the next slide, please? Yeah. So this is the content I would like to discuss today. What is the biosimilar innovative drugs for biosimilars? And the second one is manufacturing process of biosimilars. I discuss on already large scale manufacturing. I give just a brief introduction about the preclinical pharmacology. So which is mainly pertaining to the biosimilars. I don't want to give the details because it's already shared by what are the different lab animals in preclinical studies. I think what is the role of lab animals in preclinical studies? All the animals, especially the pharmaceutical industry, for filing the dossiers. But in this case, I would like to present only the preclinical pharmacology pertaining to the biosimilars. And the next part, I'll be introducing the main topic of a brief introduction about the regulatory guidelines and follow the discussion on regulatory perspectives. It is mostly the discussion on the regulatory perspective of biosimilar and safety evaluation, and finally we'll go with the question and answers. So let me start with what are the biosimilars innovative drugs definitions. Can you go to the next slide? So all the biosimilars, I think there are certain definitions of biosimilars. So biosimilars are the products which are highly similar to biological reference products, and there are no clinically meaningful differences regarding the quality, biological activity, safety, and efficacy. So this is the definition of the biosimilar, and there will be a lot of confusion about what are the generics and the generics is predominantly used in all the countries, but we cannot consider this as the biosimilars are not completely generic because of the complex nature, and they are produced mainly in the living systems, so it can never be duplicated the originated drugs, the original drugs. So that's why these biosimilars are also called as similar biological products. Just I wanted to give one generics, it is a small molecule product. It is within a single simple structure. It can be exactly reproduced. So sometimes the generics are also called as copycats.
Dr. Raju, you can stop your video so that your voice becomes more clear. Voice is breaking. I think okay.
Can you hear me now?
Yeah, now you can speak, sir.
Yeah. Should I start from the first or?
No. Continue with this similar slide, current slide.
Yeah, sure. Okay. Thank you. So just I wanted to tell about the generic drugs. So we wanted to give the generic drugs, the active ingredient in small molecule products. So which is typically a single simple structure which can be exactly reproduced. So that is what you mean by generics. But so sometimes these generics are also called as copycats. But so the biosimilars, these are the protein products, especially this protein production which involves many steps with many variables. For example, cell lines, media, cell culture conditions, purification, so which will have impact on the final product. So that's why they are having differences of glycosylation, oxidation, deamidation, etc. So generally these biosimilars, we call, I can say the process is the product here. So that's why we cannot call this as, they are not the generics, or they are not at all the copycats. There are different terms, about the biosimilars. See, some countries, they call this as in different names. For example, if you take USA, Australia, and China, so they call this as biosimilars. Suppose if you go to the EU, European Union, so this is called as similar biological medicinal product. In WHO, so they call this, they refer to this as similar biotherapeutic product. Japan and Korea, so they call this as biosimilar product. In India, we call this as similar biologic. Can you move on to the next slide? Yeah. So then coming to the innovative drugs. What are the innovative drugs? So the innovative drugs are mainly, the manufacturing companies, they produce the reference product, which is a single biological product. It's already approved by FDA, against which a proposed biosimilar product is compared. So this is the reference product. So in different, suppose if you take the FDA, they call this innovative product as a reference product. So in short word, we can call this as RP. So suppose in case of EMEA, European Union, so they call this as a reference medicinal product. So these are the different abbreviations they use for the innovative products. So RMP and RP. So all these are the original medicinal products which has been granted in the marketing authorization by a member state or commission on the basis of a complete dossier. So this is regarding the innovator products. Just I wanted to give a brief introduction about the innovator products, that is the reference products and the biosimilars. The pathway for developing the reference products. So it starts with the discovery, development, and followed by preclinical, following with clinical trials such as phase 1, phase 2, and phase 3. So all put together, if you consider from discovery to phase 3, so it may take 12 years. Then coming to the biosimilar pathway, so the development starts because there is no discovery. So it starts with the development. So these are the complex molecules, it will be developed immediately. Then followed by the preclinical and phase 1. So there are no phase 2 trials required for the biosimilars. So directly this can enter into the phase 3 trials. So all put together, so it may go to 8 years, up to 8 years it may take for the complete development process. And the complete for this biosimilars production, so the production cost may vary from $100 million to $200 million. Can you go to the next slide, please? Yeah. Just before going to the manufacturing of these biosimilars, just I would like to give more emphasis on the development pathway for the biosimilars and originated drugs. What is the difference between the biosimilars and originated drugs? Suppose if you consider a pyramid, so the base of the pyramid for the biosimilar manufacturing is the analytical part. So there is extensive analytical similarity we have to show when we are going for the market. And if you go to the top of the pyramid, the next one is the non-clinical studies. So non-clinical studies, I'll explain what are the different non-clinical studies required for the development of biosimilars to show the biosimilarity. And the non-clinical is next top of the pyramid is the PKPD studies, especially in the clinical trials which are required. And on the top of the pyramid, so the clinical, we have to show both the efficacy and safety. Suppose in case of originated drugs, so the reverse is true. So if you put the pyramid upside down, so the analytical part comes actually this is the minimum requirement, analytical for innovative drugs. But there is an extensive non-clinical studies, and followed by the clinical PKPD studies are very expensive, and ultimately the clinical studies are very expensive for the originated drugs. So this is the difference between these two. Then coming to this manufacturing of biological products, just I wanted to emphasize before going for the large scale process, just I would like to give introduction about the R&D part. So the R&D part, we have different functions in this. So what we can call this as the first one is the C&D, clinical cell line development. The second one is UPD, upstream process development, then downstream process development, and followed by analytics, bioanalytics. Then finally the formulation will come. The formulation will be there, and once the formulation is developed in the R&D setup, it will come for the preclinical pharmacology and toxicology studies. So just I wanted to give a brief introduction about the CLD part. See, in case of the cell line development and all, so whatever the reference medicinal product, the amino acid sequence is, that will be incorporated and into the gene optimization. They do the gene optimization and synthesis after incorporating the gene into the vector. So that is called as the cloning into the vector. Once the vector is done, then subsequently it goes to the host cell media. So that is, there are different host cell media. So maybe the CHO media or E. coli media. So there are different types of media. Then they pool all these things. They call this as a stable. Once the stable pools, the clones are ready. So they do the clones isolation, and they use different clones for the isolation using different techniques. For example, ClonePix. ClonePix is the one instrument which can use for the clone isolation. Then once the clone isolation is done, then they do the clone selection, because there are different clones. Out of the different clones, so they with 96 well plates, 12 plates, 6 well plates. So after that, what they do? So they prepare the cell bank. Once the cell bank is ready, then it goes to the upstream process. So the upstream process, so the upstream development, they want to screen the media and the feed. So first they culture this, the clones and all, different media. They try with different media and feed screening. And for this they'll be using different instruments such as shake flasks. So they can also use the amber equipment, because these are the 5 ml biofermenters. So this is very expensive actually. So they can do the media and feed screening. Once the media feed screening is done, and they optimize the process, complete conditions, the process conditions they are going to optimize, and they define the feeding strategy, and they do define the temperature shifts. Everything, once everything is finalized in the small media, small upstream development in small scale, then finally they select the top clone. So this final clone is called as MCB. This is master cell bank. From this master cell bank, they prove all the biosimilarity, productivity, stability, and everything will be tested from this for the master cell bank. Once this is done, then it goes to the downstream. So in the downstream process, so what is the main steps in the downstream process are? They do different types of column chromatographies. So the column chromatography, FRT chromatography, IEX, HIC, and they also purify the drug by using the ACTA purifier. Once everything is purified, so they prepare the drug substance. So this drug substance will go for extensive, analytical process. They show the analytical similarity comparing with the innovator drug, and they also prove the biosimilarity with the innovator drugs. Once everything is done, this is going for the formulation. Once the formulation is done, because all the formulations are patented, so they have these biosimilars companies. They have to develop their the formulation which is not patented, and they have to do the complete formulated drug substance which is ready for the preclinical testing. Then before going for that, can you go to the next slide, please? Once the process is optimized, so this will be, because all the R&D activities will be done in small scale. Small scale means it will be started with 5 liters bioreactors up to 50 liters bioreactors, sometimes even 200 liters bioreactors. So up to 200 liters we can do it in the small scale. Once everything is stabilized, the process is stabilized, then we transfer this to the manufacturing team. So the manufacturing team, again, they have the upstream process, downstream process. So in the upstream process, the drug substance, what they prepare, they use from the working cell bank. So this is they use for the propagation. And after that, the downstream, so they purify the process to remove all the impurities. Once all the impurities are removed, then it goes for the formulation. So this is then we call this as the formulated drug substance, or we can call this as a drug product. So this is mainly to increase the safety and stability. So there will be more efficiency for this. Once the formulation, the drug is formed, then it will be going for the filling and packaging for convenient usage. So this may be filled in either in vials or pre-filled syringes. Then probably after completion of this will go for the storage. So this is the complete process of biosimilars. From development of biosimilars from R&D to the large scale. Can you go to the next slide, please? Because I don't want to give all the details of the manufacturing process because this is not of our interest. Okay. Then coming to the preclinical pharmacology, introduction to the preclinical pharmacology. The preclinical pharmacology means, suppose if you take the modules. So module 4, I think you are familiar, all the people are familiar with the module 4. So which is the part of the dossier submission. So which involves the in vitro activities, in vitro pharmacology we have to take, and the non-clinical studies which also involve the in vivo part, the pharmacodynamics. So the preclinical pharmacology is mainly stepped into 4 aspects. The first aspect is in vitro pharmacology to show the mechanism of action. The second one is the in vivo pharmacodynamic activity to go for the animal model development to show the efficacy, and the pharmacology, the other pharmacology part is the pharmacokinetics. So once we complete pharmacology, then we go for the toxicology. Even with one particular thing, especially in the biosimilars, what we are supposed to do is the immunogenicity, because these are the foreign proteins which are highly immunogenic in animals or in humans. So the mandatory requirement is the study of the anti-drug antibodies. So we'll go into the details of the preclinical pharmacology. I told so the structure, especially if you go for the characterization. So there will be 2 kinds of characterization. One is the physical and the second one is the biological. So we have already discussed there is an extensive structural and functional characterization is mandatory for all the biosimilars. I told the pyramid, the base of the pyramid is analytical. So this step is very extensive in case of biosimilars, where it is not necessary for the innovator drugs. Because this analytical similarity, this is mainly to understand the molecule and its function, and we have to identify the critical quality attributes which are clinically active components. Then, so the extensive structural characterization using various analytical techniques, various analytical techniques studying the analytical properties, especially the primary and the higher order structures. So the second part is the appropriate quantitative biological assays we have to do the functional characterization. Can you go to the next slide?
Sorry for the inconvenience first.
Can I proceed?
Yes, please continue.
Yeah. So I told you that the first part is in vitro biological activity, in vitro PD. So just I want to give some examples of, I want to take a reference of monoclonal antibodies. Say I'm not sure because the people are familiar with all the terminologies of biosimilars. The biosimilars, sorry, I have not introduced those, what are the different biosimilars earlier. So there are different, these are recombinant proteins mostly. So they are also called as therapeutic proteins. They can be divided into different categories like therapeutic proteins, fusion proteins, and monoclonal antibodies. So, for example, if you take the therapeutic proteins, filgrastim is one example, erythropoietin, darbepoietin. So these are the therapeutic proteins. Suppose if you consider the fusion proteins, all the septs like abatacept, etanercept. So these are the biological molecules, especially the fusion proteins. Suppose if you are referring to the monoclonal antibodies, now entire world, most of the biosimilars, especially 75% of the biosimilars are the monoclonal antibodies. So the monoclonal antibodies, not only the biosimilars, even the reference drugs. So the monoclonal antibodies, for example, rituximab, trastuzumab, all the maps, trastuzumab, rituximab, even bevacizumab, So these are the different infliximab. So these are the different monoclonal antibodies. Just I want to give a brief introduction about what are the in vitro studies. Suppose if you consider the monoclonal antibody as an example. So the monoclonal antibody, you can see the figure. So there are all the antibodies. So mostly they are mostly the monoclonal antibodies are from the IgG1, which contains the Fc portion and Fab portion. So there are two Fabs you can see. So the Fc portion and the Fab portion. So the Fab portion is mainly mediated mechanism of action to indicating the efficacy. For this, there are different endpoints we can use, the different cell lines. We can go for the proliferation, neutralization assays we can do, receptor activation, cytotoxic assays, and signaling assays to understand the complete Fab-mediated mechanism of action to see the efficacy. So all these assays are cell-based biological assays. And we can also, for the Fab-mediated method, to understand the Fab-mediated mechanism of action, we can also go with the target binding assays. So the target binding assays are also mainly the cell-based or ligand-based assays. So these cell-based assays and ligand-based assays, so they can be done either by using, especially by ELISA, or you can use certain instruments such as SPR. Surface plasmon resonance. So these are the for binding, especially for the binding. Suppose if you want to go for the Fab and Fc mediated mechanism of action to see the efficacy, we can take different endpoints like ADCC. Like ADCC is antibody dependent cellular cytotoxicity, CDC complement dependent cytotoxicity, and ADCP. This is antibody dependent cellular phagocytosis. So these are the different endpoints we can take. For this also we can use different cell-based biological assays. Suppose if you want to take Fc portion, Fc binding assays, there are different Fc binding assays. For example, if you take Fc gamma R binding assays, even Fc gamma R also there are different fragments we can go. Fc gamma R1, R2, R2A, R3. So different Fc gamma receptor binding assays we can do. These are the different ligand binding assays, and we can also use the complement binding assays to see the efficacy, which is mainly safety indication of Fc binding. And some of the functions like Fc binding assays, especially for the indication of the PK, FcRn is the main endpoint. FcRn binding is because the monoclonal all the monoclonal antibodies, mostly they are expressed in higher animals. Especially we can say they are the relevant species are the monkeys, cynomolgus monkeys. But if you see the cynomolgus monkeys, we cannot do all the in vivo studies in cynomolgus monkeys because of the restriction of usage of animals. See some of the functional activities like FcRn binding and all. So this FcRn is also expressed in case of rodents, especially rats and mice. So which is mainly mediated the clearance of, especially in the PK, the clearance of the drug, especially the monoclonal antibody, it is mainly mediated through FcRn binding. So this can be done in the in vivo studies in rodents for monoclonal antibodies just by using this ligand binding assays. So this is a nutshell different cell types, different biological activities, especially to show the biosimilarity, in vitro mechanism, to show the in vitro mechanism of action. These are the different assays we use. Then coming to the in vivo pharmacodynamic activity. So once the in vitro pharmacodynamic activity is completed, then we'll go for the in vivo pharmacodynamic activities. I told you. So the in vivo pharmacodynamic activity, as per the guidelines, so this is because of unnecessary usage of animals, because restriction of usage of animals. So this is not a mandatory to go with the in vivo pharmacodynamic activities in all the cases. For example, if the relevant species is not rodents or non-rodents, if the relevant species is only monkeys, so we cannot use any in vivo monkeys for conducting the in vivo pharmacodynamic studies in monkeys. So that's why this is an optional case. Suppose if you show the functional similarity and biosimilarity of the molecule, and if you could establish a complete the mechanism of action by showing the in vitro assays, then obviously there is no absolutely need for going for the in vivo pharmacodynamic studies. So this is the companies, pharmaceutical companies, they go with the in vivo pharmacodynamics. Why? Because to take the comfort factor. So they wanted to include this to substantiate the studies to what they have done with in vitro to show the functional similarity and biosimilarity. Suppose in case of in vivo assays, which include the animal models of the disease to evaluate the functional effects on the PD markers or efficacy measures. So for example, I can give you one example of in vivo assays, especially developing of animal models. If we take pegfilgrastim or filgrastim, so this is the main function of the filgrastim or pegfilgrastim to increase the neutrophil counts. In case of neutropenia. So this drug is mainly for human beings. This is given as an adjuvant therapy to the patients who suffer the cancers. So any cancer, if you give any anti-cancer drug, so they start with the filgrastim treatment because so all the chemotherapeutic agents, they suppress, not only suppress the normal cells, they also suppress the other, not only suppress the cancer cells, they also suppress the normal cells. Especially the first organ which is mainly affected is the bone marrow. So, for example, if the bone marrow is suppressed, there will be complete loss of neutrophils. Because this condition is called as neutropenia. Because of the treating the cancer patients with anti-cancer drugs, so there will be always the side effect called as neutropenia. Because of the complete neutropenia, so this is called as referred to as febrile neutropenia, because neutrophils are the main target, the main self-defensive mechanism in case of combating the diseases. So the patients, the cancer patients which are who are undergoing anti-cancerous therapy, they are more prone for the infections. So the next infection is because of the infections, it causes the febrile neutropenia. So this condition is the worst condition. So that's why especially the filgrastim. So this is nothing but the granulocyte stimulating hormone. So this is a recombinant product. So which can induce the neutrophils in the bone marrow. So to increase the neutrophil count. So this is the indication for filgrastim, which can be used as an adjuvant therapy in human cancer, especially human anti-cancer, in cancerous patients while undergoing chemotherapy. Because the same kind of condition in animals also we can create. So we can induce, for this we can select mice. We can induce the neutropenia by giving some anti-cancer drug, like you can take cyclophosphamide. You can use the cyclophosphamide to animals. And once we give that cyclophosphamide to the animals, within 3 days there will be complete severe drop in the neutrophil count. And after this, maybe on third day, if you give the pegfilgrastim on the day 3, so it takes 3 to 4 days to develop the neutrophil count. And if you see after 6th day, so there will be exaggerated, the increase in the neutrophil counts. So this is the main. So the objective is to increase the neutrophil counts. So here we are creating the condition neutropenia. After condition is created using the anti-cancer drug, we can introduce pegfilgrastim or filgrastim drug, and thereby it increases the neutrophil count. So always you remember any activities, especially for the biosimilars, for the in vitro activities or functional similarity, analytical similarity, biosimilarity, everything, always we have to compare with the innovator drugs. So this is the important pharmacodynamic activities. So this is the one model that just I am representing for the pharmacodynamic studies. Next part is the pharmacokinetic studies. The pharmacokinetic studies for large molecules is completely different, because all the large molecules, especially if you take the monoclonal antibodies, they have the longer half-life. For example, these can be administered either via subcutaneous administration or intravenous route. It depends on the type of the route of administration in human beings. So the human beings, what is the route of administration? The same route can be administered in animals to see the pharmacokinetic profiles. For this pharmacokinetic evaluation, so there are different steps. So before going for the analysis, they do the pharmacokinetic development. So there are different pharmacodynamic, there are kinetic developments. So they do the development and followed by the validation. So once the development is completed, so they do the validation, especially to see the accuracy, precision, sensitivity, selectivity, specificity, and they also check the dilution, linearity, and stability. Once everything is developed and validated, and this is ready for the analysis. For example, if you take any pharmacokinetic studies, we can administer the drug. Suppose if you take any monoclonal antibodies in animals, especially rats or mice. So generally we use the rats even for monoclonal antibodies because even though they are not the relevant species, because this is mediated by the FcRn mediated elimination. So we completely use this rats or rodents for the analysis of pharmacokinetic parameters. So if you give the drug can be administered either by subcutaneous or intravenous route, and at different time points we collect the blood, separate the plasma, and give it for the bioanalytical team. So they do complete analysis of the methods are already validated, developed and validated. Using that validated particular method, so they'll be conducting complete analysis of the samples, and it will be incorporated using the Vinolin software. So we can do complete analysis of pharmacokinetic. We can do the complete analysis of the pharmacokinetic parameters. Yeah. So the next part, yeah. So the main important thing is, along with this pharmacokinetic parameters, the next parameter we generally do is the immunogenicity. So there are this immunogenicity, especially the large molecules as monoclonal antibodies. So they are highly immunogenic. They can induce immunogenic response when administered to either healthy or diseased population subjects. So here is we have an approach for the clinical samples, how we are doing, but the same type of approach we can also go for this preclinical sample analysis. So there are different kinds of there are 4 tiers: tier 1, tier 2, tier 3, and tier 4. So these are the 4 tiers especially used for screening of the samples and do the analysis of anti-drug antibodies. So the first one is the screening assay. This is the tier 1. Tier 1 is the screening assay. So this will be used for the anti-drug antibodies. First they screen the samples. Of course, this is mainly designated for 5% false positive. Suppose if the samples are negative, then there is no because they are not immunogenic. And if the assays are positive, if the screening is positive, then they go for the confirmatory assays. So this confirmatory assay establish to true anti-drug antibodies designed for that particular product. So there may be 1% false positives. Once the confirmatory assay is done, then they go for this. Suppose if any positive result comes, then tier 3 they go for the titer assay. So they check the titers in the samples. Suppose in case of tier 4, so up to tier 3, tier 1, tier 2, tier 3. So these are common assay methods and procedure for the preclinical samples. But in case of clinical samples, they go further one more step. So they go with the tier 4, and this is called as the NAB assays, neutralizing antibody assays. So this is also mainly meant for 1% false positives. So then suppose if they find positive in the NAB assays, and so these conditions are called as neutralizing antibodies. They report this as neutralizing antibodies, and if it is negative, then they report this as non-neutralizing antibodies. So this is the complete screening picture of the immunogenicity in case of preclinical and clinical samples. So this is the additional thing which is not there for small molecules. Can you go to the next slide, please? Yeah. Okay, so we have completed so far the preclinical pharmacology, starting with the in vitro mechanism of action, in vitro biosimilarity using the bioassays, then in vivo pharmacodynamic studies, followed by PK pharmacokinetics, and the special concept on anti-drug antibodies. So these are the different sections in the dossiers. Then coming to the introduction to the safety evaluation. So once the pharmacodynamics, pharmacokinetics parameters are completed and the drugs show, if you show the biosimilarity, then the toxicology is the main component, which is a mandatory for before going for the clinical trials. Can you go to the next slide? Yeah. So, in the toxicology, the preclinical studies have always been a critical component in the development program of biopharmaceuticals. So this is a mandatory drug. So I think, Dr. Siva, in the last webinar, so there were extensive toxicology models. What are the different studies? So we have already, already about those different kinds of studies. But all the studies may not be required for the biosimilar, especially for the development of the biosimilars. A simple study, a 28-day comparative repeated dose toxicity studies, which involves both the TK, immunogenicity, and PD marker. So that is sufficient to go with the next clinical phase filing. So, for example, if you take the local tolerance study, which is a mandatory for all the small molecules, so we can separately conduct the local tolerance study. But this local tolerance study also be clubbed with 28-day repeat dose toxicity studies. So we can say if you conduct one repeated dose toxicity study, where we can include the local tolerance, the TK profiles, and PD marker analysis, immunogenicity. So with all these matters, we can completely do one complete study on the repeated dose tox study. So based on the repeated dose tox study, we can find the dossiers. For example, just I wanted to give one example for the comparative repeated dose toxicity study in rodents. For example, if you take, all the toxicity studies as per the biosimilarity guidelines. Of course, I'll talk a little bit about biosimilarity guidelines in subsequent slides. The relevant species, unlike the small molecules, the difference between the small molecules and large molecules is because the receptors are expressed only in certain group of animals. So those animals should be considered for conducting the toxicity studies. So suppose if you take the therapeutic proteins, for example, filgrastim, pegfilgrastim, even darbepoetin. So for these molecules, the relevant species is rodents. Even the receptors are expressed even in the rodents. So we can select one of the rodent molecule species for to conduct the repeated dose tox study. For example, so this is a typical study design, which especially in India, suppose if you go with the RCGM and all for biosimilarity guidelines, so they have mentioned, so we have to take one control group, so which can be used for giving the formulation buffer. The second group is the biosimilar groups. So, but in the biosimilar groups also we have to go for 3 dose levels. So I want to say 1x, 2x, and 5x. These are nothing but the human equivalent dose. Suppose we know all the molecules, the human dose. For example, if one human dose is 1 mg per kg body weight. So suppose if you consider in animals, based on the metabolic rate. So there are different. We have to convert the human equivalent dose in animals. Suppose if you take Vistar rats. So the human equivalent dose will be 6 times because the metabolic rate is more 6 times more when compared to the human. So we can take 6 mg per kg body weight is the 1x dose, 1x human equivalent dose. So 12 mg will become 2x human equivalent dose, and similarly, 6 mg, 12 mg 2x, and 30 mg per kg body weight will be the 5x of the human equivalent dose. And coming to this, but we can keep one reference, because even for toxicity studies also, so we have to keep one reference drug group. So the group contains we can take only 1x human equivalent dose. So we can go with 1x, unlike 3 different dose levels. So we can go with the 1x human equivalent dose. So that is the main group. So we can also keep the recovery group of animals, especially for the control group and biosimilar high dose group, 5x group. And so the repeated dose tox studies, if of course, if it is a chronic tox study, if it is for the monoclonal antibodies, so we can go for up to 26 weeks. But even as per the guidelines, so we can restrict the study to 4 weeks. So we can give the drug either on day 1, day 8, day 15, day 22, and day 29. And all the parameters what we are looking into that is clinical signs, body weight, food consumption, and all the other parameters like clinical chemistry, hematology, coagulation, clinical chemistry, urinalysis, bone marrow smear evaluation, organ weights, gross pathology, histopathology. So all the parameters we have to analyze. And apart from that, we also look for the PK and ADA. We can collect the samples. We can collect the samples on day 1, day 8, day 15, day 22, day 29. So for the anti-drug antibody analysis, suppose if you want to include the PK dose also, we can go with the sparse group of animals, additional animals in each group. Then according to the time points, we can use those animals for the collection of the blood for the PK studies. And this is once on day 30th, we can sacrifice those animals for the necropsy. And if it is for recovery group of animals, especially the control group and the high biosimilar dose group, so 43rd day we can go for the recovery group necropsy. And again, we have to go for the hematology, clinical chemistry, organ weights, so everything, even at the and towards the end of the necropsy, both for main and recovery groups. So this is the nutshell actually the toxic design. Suppose if you consider the RCGM or India for the India dosing filing. Okay. Then, so this is the pre-introduction about the preclinical pharmacology and the toxicity. Then we'll go to the regulatory. So this is the main topic is the regulatory guidelines. What are the different regulatory guidelines we have? Can you go to the next slide, please? Yeah. So the regulatory guidelines, so if you consider the regulatory guidelines, so in the entire world we have main 3 regulatory guidelines. One is mainly followed by the European Union, the second one is by United States of America, USA, the third one is the global. So these are the either regulatory guideline or guidance documents. So generally we follow, to consider for dossier filing in the entire world, But if you say, suppose if you take, the package ICH S6R1, so this is mainly used for the novel monoclonal antibodies and other for innovative drugs. But if you consider the biosimilarity guidelines, mainly the European guidelines, so it can be divided into 2 aspects. One is the EU guidelines on similar biologic products. We have separate guideline for that, and similarly we have a separate guideline for on biosimilar medicinal products, especially containing the monoclonal antibodies. So we can refer to these guidelines, non-clinical and clinical issues. Suppose if you consider the United States of America, USA, so there is scientific considerations in demonstrating biosimilarity to a reference product is the guidance document we have. And suppose if you consider the other guidelines, the major guideline is WHO guideline on evaluation of similar biotherapeutic products. So these are entire world either they follow these guidelines, either European guidelines, some countries they follow the USA guidelines, but some majority of the rest of the world, so majority of the guidelines they follow. I mean, majority of the countries they follow the WHO guidelines. But based on the WHO guidelines, can you go to the next slide, please? Yeah. So based on the WHO guidelines, because the rest of the world they follow the WHO guidelines. For example, if we take China, so they have a guideline based on the WHO guidelines. They have they have a guidance document, guidance for research and evaluation techniques for biosimilars. This is the guideline for China. Canada, they have a guidance for sponsors, information submission requirements for subsequent entry biologics, SEBs. Japan, they have a separate guideline, PMDA guideline for quality, safety, and efficacy assurance on follow-on biologics. And India, we have a similar biologics regulatory requirements for marketing authorization in India. So these are the different guidelines, but they follow the WHO guidance document. Can you go to the next slide? Can you go to the next slide, please?
Dr. Ratnadeep, next please.
Yeah. So the next part is, see, after introduction of the regulatory guidelines, then the basic topic is the regular discussion on the regulatory perspective of biosimilarity assessment and safety evaluation. You know, can you go to the next slide, please? Yeah. See, different. We have already discussed about different guidelines. So what exactly the guidelines says? So suppose if you take the EU regulatory guidelines, so they completely provide a path that allows the development and operation of biosimilars with the submission of non-clinical in vitro data alone in certain circumstances and without non-clinical in vivo data. So what does it mean? Suppose if you want to file a dossier in European countries, so we can go with the in vitro data alone. So there is no requirement of non-clinical in vivo data. For example, they don't require the PD marker, pharmacodynamic studies, in vivo pharmacodynamic studies, and they don't even consider the toxicity studies for this regulatory filing, especially in the EU regulatory guidelines. But provided you should have complete biosimilarity and functional similarity, should say there are no differences between the biosimilar molecule and the innovator molecule. So this is the mandatory thing. But suppose if you show complete functional similarity and biosimilarity in EU region, then there is no requirement of any in vivo studies or any toxicity studies. They can directly go for the clinical dossier. So we can submit the clinical dossier without the non-clinical in vivo data. Then suppose if you take the US guidance document, so this is a little bit trickish. So they follow a stepwise approach. So that means if you show the complete biosimilar, if you show any differences between the functional similarity and in vitro biosimilarity aspects, then they may request, the guidance document says, so we should get, at least we should discuss with the regulators, especially the US regulators, and you can go with simple toxicity studies. So that's why we call this as a stepwise approach. See, actually, suppose if you show complete biosimilarity and analytical package, which is almost similar to the innovator drug without any significant difference, they can also ask for waiving of the in vivo studies. But this is not possible for the biologics, especially. At least we can have some differences. We have already discussed about the glycosylation, sialic acid content, amidation. So there will be differences, especially in the structure and functional activities. Obviously, they ask us to go with at least one study, but it depends upon the extent of the differences. So they may ask for complete toxicity studies. We have to go with the complete toxicity studies using different dose levels, and we have to compare with innovative drugs. And we have to go with all the clinical parameters. We have to go with for the tox studies. Otherwise, they may also ask for, we need not go for complete repeated dose tox studies. They may ask us to go with the PK parameters, just the comparison of the PK parameters and immunogenicity. So it depends upon the type of suggestion. When suppose if you go with the FDA guidance with the team, so they may refer us to go with the complete repeated dose tox study, or we may go with the simple TK and ADA study in the animals. But the next approach is the WHO guidance document. So this describes the non-clinical. Go to the previous slide. Can you go to, yeah. So this WHO guidance document describes the non-clinical evaluation of similar biotherapeutic products. So irrespective of whether you show the functional similarity or analytical similarity, they request us to go for at least one repeated dose tox study. So that's why suppose if you take all the all the guidelines, EU guidelines says it is tox studies are not required or in vivo studies are not required, but US says it's a stepwise approach. The stepwise means if you show the complete similarity, so we need not go with the tox studies. But if you don't show any complete similarity and biosimilarity with innovative drug, then obviously we have to go with the. So this is the moderate approach. But the third WHO guidelines, which clearly mandates that one is at least one repeated dose tox study, which is mandatory for going for the clinical dose submission. So, but rest of the world, many countries they follow WHO guidelines. So that's why this guidance document they took this WHO guidance document, but they interpreted in such a way that which is suitable for different countries. We'll go to the next slide, please. Yeah. So many countries they have established their national guidelines that appear to be based on the WHO guidance document. For example, India, Brazil, Republic of Korea. So they have their own. But the interpretation of so WHO said only one repeated dose tox study is required, but they have not clearly defined the structure how we have to do that. But based on that, different countries they have established their own guidelines. For example, if you take the Brazil. So the repeated dose tox study is a mandatory thing that should be conducted in relevant species. I told you, the relevant species is the receptors have to be expressed. Unfortunately, many of the monoclonal antibodies, most of the monoclonal antibodies, the receptors are mainly expressed in cynomolgus monkeys I can say. So obviously we have to choose the monkeys for all the toxicology studies, repeat dose tox study. So we can conduct the study as per the Brazil guidelines. We can conduct the study, repeat dose tox study for 4 weeks, but we should include the TK parameters and ADA parameters as a part of repeat dose tox study. Suppose if you consider India also the repeat dose tox study is a mandatory in relevant species, which is a 4-week study. ADA is a mandatory, but TK is not a mandatory thing in biosimilarity guidelines in India. But we should take 3 dose levels here. 3 dose levels means we have to go with 1x dose, 2x dose, and 5x dose. And obviously we have to increase the. We have to include the recovery groups as well. So control group recovery and high dose group biosimilar recovery should be kept as a part of the design. Suppose if you take Japan. So they follow mostly the ICH ethics, and they also go with the stepwise approach. But Japan, if you consider the Japan guidelines, so they said so the relevant species are not necessary. Even repeated dose tox study can be done in non-relevant species. So that is the exclusion in case of Japanese guidelines. But Australia guidelines, so Australia they follow the EU guidelines, and China they follow WHO guidance document. And they also say we have to include the PD marker. So there we have to include a PD marker, PK in the comparative repeat dose toxicity studies. But at least we should use the low dose and high dose. So the middle dose 2x dose is not required for this. But it should be conducted for 4 weeks, and TK and ADA are also part of the requirement. See, the interpretation of WHO guidelines is very different in different countries, but the challenge for for the pharmaceutical companies, especially in our country, so we are facing a lot of problems on this, especially to have one tox design. For example, if you take, I'll go with one case study by this. I want to discuss about the monoclonal antibodies, the tox studies. I told you, so the monoclonal antibodies with the relevant species is cynomolgus monkeys. But you can refer to that one article. So this is Chapman et al. So they conducted a survey with the different CROs, especially for the monoclonal antibodies development and all. So this was published in the MAPS journal. So according to their survey, so 75 almost they conducted 26 studies survey for 25 products, and they said that most of the studies, especially for the monoclonal antibodies, 75% they used cynomolgus monkeys in all the studies. And if you consider the male and females, especially 3 males and 3 females, they selected for each group. And most 75%, that's also 75%, and 45%. So they used 1 high dose of the biosimilar and 1 high dose of the innovator drug. And 24% of the studies, they had two dose levels. One is one low dose and two high dose groups they used for biosimilar and innovator drugs. Then 95% of the companies, they have not kept any recovery groups, 95%. But all the studies they conducted for 4 weeks. So this is as per the article. So this is the reference what they have given. And the most common study what they have used is 3 males, 3 monkeys, 3 males and females for each group, and they used 2 dose groups, one low dose and one high dose group, and they conducted studies for 4 weeks, and mostly there are no recovery groups. So this is the type of the tox studies as per the for biosimilars what they have conducted as per this reference article. Can you move on to the next slide, please? Okay. Then, so based on the guidelines, what is our approach? The pharmaceutical companies, all the pharmaceutical companies, we file the dossiers in different countries, because it may be in India, it may be China, Brazil, EU, FDA, because there are different guidelines, different designs and all. So what could be the common design we can use, especially? So for example, I want to take the example for monoclonal antibody. What is the common design? What we are preferring? Of course, we can have lot of discussion on this. This is a comparative repeated dose toxicity study of a biosimilar mAb versus reference product. So always we have to consider the reference product or reference medicinal product, because for dosing the file, especially for filing the dossiers, always we have to compare with the innovator drug. But the innovator drug has to be registered in that particular country. So always we should compare with that particular country. See, in the world, especially 2 innovated drugs are being used. One is marketed in EU and one is marketed in US. So that's why I want to call this as the reference product, because it's a reference product is mainly in US and RMP is in EU region. So always, because we cannot conduct separate studies exclusively to file the dossiers either in Europe or in US. Obviously, what we have to do, we have to conduct the study, we have to compare with both the innovator drugs. One is with the reference product, the other one is reference medicinal product. For example, this is the design for subcutaneous administration for 4 weeks to cynomolgus monkeys. So the doses, if you see the doses, one is the first group is the control group, so which can be exclusively used for the formulation buffer. The second one, biosimilar mAb, so we can consider 1x dose. And the third group is the biosimilar mAb. We have already discussed about the human conversion dose and all. I don't want to further detail on that. Biosimilar mAb 5x dose, and similarly we have to compare with reference product 1x, reference product 5x dose, RMP 1x dose and RMP 5x dose. Suppose if you consider, so we can also put the recovery groups, because we have already discussed in India. So the requirement of recovery group is important, it's a mandatory thing. So that's why we can keep two recovery groups, one for the control group, the second one for the high dose of the biosimilar mAb, and the other groups are not required as per the design. So this is a nutshell the design of especially for monoclonal antibodies in cynomolgus monkeys. So the injection frequencies what we can give is on day 1, day 8, day 15, 22 and day 29 we give. Because since it is subcutaneous administration, 1 ml per kg body weight we can give the dose. And the study, total duration of the study is 43 days. So necropsy for the main group of animals will be conducted on day 13. And necropsy will be conducted for the recovery groups on day 43. And TK sampling on day 1 and day 22, so we can use the same animals for collecting the blood. So we can go with the day 1, day 22 at predetermined time points. So in between also we can take the bleeding on day 8, 15 and day 29 also. And observations, of course, the next slide will talk about the complete details of the observation, how we are using this study. Can you go to the next slide? Yeah. So these are the different parameters what we are looking for. So suppose if you take the cynomolgus monkeys, there will be a pre-treatment period. So the animals are acclimatized, randomized, and after randomization, they use it for 7 days to 15 days is the time period gap. So they complete the pre-treatment assessment they do. For example, ophthalmology, electrophysiology they do. That's the ECG. Blood pressure they measure. Clinical pathology. So disease analysis complete. Sorry, this is urine analysis. So this is the complete picture will be in the pre-treatment period. Once they administer the drug on day 1, and before administration of the drug, they collect the samples for AD and TK. These are the pre-dose samples, and they administer the drug on day 1, day 8, day 15, day 22, and day 29. And during the period on day 1, day 8, day 15, day 22, day 29, so they collect the samples for ADA, and at different time points also the PK samples will be collected, analysis of the PK parameters. Day 20, especially during the week 22, day 22 or week 4, we can say the ophthalmology, everything will be completed in that part. And the day, before day 29, so they will be conducting the electrophysiology, especially ECG, blood pressure, and everything will be monitored. And on day 29th, we can sacrifice, day 30th, they sacrifice the animals, and they collect the blood. So for processing clinical chemistry, bone marrow evaluation they do, complete clinical pathology, so bone marrow evaluation, organ collection, and histopathology, everything will be done on day 30th and day 43 for necropsy group of animals. So this is this is the design of the experiment what we can propose for global submission. Now it's the time for question and answers.
Thank you, sir. Now I request Dr. Bhasa sir to start the question and answer session.
Thank you, Dr. Ratnadeep. Till I have received only one question. One question from my side is that sir, is there any issue related to immunogenicity and the drug has to be withdrawn? Are there any examples like that?
Yes, doctor. See, actually, the immunogenicity part, anyhow, we are comparing with the innovator drug as a part of toxicity study. Suppose if the animals are immunogenic for the biosimilar drug when compared to the innovator drug, obviously we are not going for crossing the drug for the clinical trial.
Okay. One question from is that are there any other companies other than Reddy's Lab who are working on the biosimilars in India?
No, Dr. Reddy's, we have only one unit on biosimilars actually, biologics. This is called the biologics department.
Okay.
So complete starting with R&D till manufacturing. So completely it's under one roof.
Okay.
It is in Hyderabad.
So are there any other companies in India who are working on biosimilars like Reddy's?
Yeah, there are many companies like Biocon, you have Aurobindo, they have biosimilars, Cadila, every Vocard, Intas. So all the companies they are working on biosimilars.
Okay.
Seems a bright future for the biosimilars or biologics as compared to.
Yeah.
Dr. Raju?
Yeah.
What is the future?
Yeah. So the future, even mostly all the pharmaceutical companies, they are coming with biologics. So the next future is the biologics or biosimilars. Because biosimilars, they are mostly investing on the biosimilar plants.
Okay. One question, Dr. Ramesh is there. Which is the common technique used for concentration analysis, since these are all proteins?
Yeah. So generally, all the proteins will go with only one technique. We can because HPLC and all we can use for this one quantification. Generally, we use the spectrophotometer UV280 for concentration.
Okay. One more question is that which are the assays which are preferred for anti-drug antibodies?
Sorry, can you repeat the question because the voice is breaking?
Which are the assays preferred for anti-drug antibodies?
So you are saying measurement of anti-drug antibodies.
Yeah.
See, actually, the anti-drug antibodies, we have a complete development. So, like PK Kinetics, so we can go with the development of anti-drug antibodies, especially once the anti-drug antibodies are developed, and this will be validated. And we can use, there are 3-4 techniques we can use, like ELISA can measure, and we can also go with the mesoscale development. This is called as MSD platform. The third one is the gyro. So these are the 3 platforms we go with this.
Okay. One more question is whether only IgG are considered as a part of immunogenicity in preclinical studies, or antibody specific to antigen, or the product has to be measured?
No. So actually, this is anti-drug antibodies only we measure as a part of this, because so actually we cannot interpret whatever the anti-drug antibodies in the preclinical sample. We cannot extrapolate the readings, especially the results to human, because even the guidelines also says we cannot extrapolate these results. So this is just to find out the TK profiles. Is there any difference in the TK profiles because of the anti-drug antibodies? For that purpose only we measure this.
Okay. What is interchangeable biologics?
Yeah.
Requirement for preclinical and clinical development.
Yeah. No, these are interchangeable biologics. The concept is completely different. Actually, once the drug is manufactured and authorized, going for the authorization, commercial authorization, so they give the permission as whether it is a similar biologic drug or interchangeable. For example, if you take interchangeable biologic means we can, without any prescription, so suppose with the prescription, so the pharmacist can change the drug. Suppose one doctor prescribes infliximab, that is the Remicade is the innovator drug. So they can give the other substitute like so they can change and give the drug, but it should be given by the commercial authorization. So then only it's an interchangeable drug. Otherwise, what especially the US and the EU region, suppose if it is registered as a similar biologic, only the biosimilar, so unless it is prescribed only the biologic, we cannot interchange it. We cannot substitute for innovator drug.
Okay. Last 2 questions, sir. What should be the percentage of similarity between the innovator drug and biosimilar drug?
So it's a difficult question to answer. So similarity means there are different similarities are there, whether it is the analytical similarity or the function similarity, or we don't consider the toxicological similarity. So almost the definition of the biosimilar says even though it's dissimilar to the innovator drug, but functionally it should prove the clinical efficacy, safety. So that is the main importance. Suppose if there is a difference, but if it is safe and if it is efficacious, then there is no question.
Okay, sir. Last question. Is dosing regime for toxicology study once in a week or similar to the human dose?
Yeah, it depends on the thing. Actually, generally it is required one dose, especially once a week is sufficient. But in some cases, if it is recommended in the product monograph, if the tox studies they have given twice a week, we can go with the twice week. But 4 weeks is sufficient.
Okay, thank you very much, sir, for answering the queries from the participants. Now I request Dr. Thakur to make the concluding remarks, then I will propose the word of thanks. Dr. Thakur, please.
Hi, good afternoon, all of you. A very nice presentation by Dr. KVS Narayan Raju. And this is a developing field, and many, as he has narrated very precisely about the regulatory toxicology as well as the clinical studies and hindrance, etc., and future of these biologics in India as well as in the foreign countries. And as he has mentioned in the introduction session, that many companies in India are working on these biologics. So there is a bright future for all the youngsters to venture this type of field. So very concise and in-depth information has been shared by Dr. K. Narayan Raju. I sincerely thank him and his the organization for permitting him for this nice presentation for the benefit of our youngsters and the participants, ICPT life members. So once again, I thank him, and his email ID is already mentioned. So all of you, please take all the benefit from Dr. KVS Narayan Raju. He'll be happy to share you the information, whatever is required. I also thank all the participants for sincere active participation, and all the particularly committee members, executive committee member, as well as for this webinar committee, Dr. Bhavsar, Dr. Ratnadeep, in absentia Dr. Srinivasan and P. Sriram. So once again, I thank all of you, please. Thank you. Dr. Bhavsar, please.
Yeah. So already Dr. Thakur has proposed a lot of thanks. So once again, from the society, ICPT, we are thankful to Dr. KVS Narayan Raju for a nice talk, as well as the Reddy's Laboratory for permitting him to deliver this talk on a very specific topic on biosimilars. And we are also thankful to all the senior pharmacologists and toxicologists